Okay, everybody, welcome back. Last year I had the opportunity to attend the UMDF Symposium in Bethesda, Maryland. While there, someone pointed out to me our next speaker. Now, some people get starstruck when they see a famous actor, athlete, or politician. Me, I was thrilled to be in the presence of the man who founded the field of mitochondria, who found the first mitochondrial mutation, which happens to be the one my family carries, LHON 11778. I mustered the courage to introduce myself, having that same awkward feeling we all have when approaching someone famous and important. Imagine my delight when not only was he willing to speak to me, he was genuinely excited to share his vast knowledge with me, someone who was interested in the topic and from a layman's term, not from the scientific perspective. What I remember most vividly was the complete passion in his statement that before he eventually retires, he wants to cure someone affected by LHON. As we all know, that's a goal we're all deeply invested in helping him achieve. Let me share with you his bio. Dr. Doug Wallace is the director of the Center for Mitochondrial and Epigenomic Medicine and the Michael and Charles Barnett Chair of Pediatric Mitochondrial Medicine and Metabolic Disease at the Children's Hospital of Philadelphia. He has a PhD in microbiology and human genetics from Yale University in New Haven, Connecticut, and he did an NIH postdoctoral fellowship at the Department of Human Genetics at Yale University. His research interests focus on the role of mitochondrial energy in human health and disease. More than 35 years ago, Dr. Wallace and his colleagues founded the field of human mitochondrial genetics. The mitochondria are the cellular power plants, organelles that generate most of the cell's energy. The mitochondria also contain their own DNA, the mitochondrial DNA, which encodes the wiring diagram for the cell's power plants. Dr. Wallace showed that the mitochondrial DNA is inherited exclusively from the mother, and that genetic alterations in the mitochondrial DNA can result in a wide range of metabolic and degenerative diseases, as well as being important in cancer and aging. Dr. Wallace has authored numerous papers, and he is a member of 11 national societies. In 2010, the National Academy of Sciences recognized Dr. Wallace with an elected Member of Institute Award. In 2006, he received an award from the Chinese Academy of Sciences as an Albert Einstein Visiting Professor, and he received the Robert J. and Claire Pizarro Foundation Award for contributions to cardiovascular disease. Please welcome Dr. Doug Wallace.
Well, thank you very much for being there, and thank you for inviting me to join you all here. I'm humbled to follow the first two speakers because they are the world's preeminent clinicians in this field. So I'm going to try and address some of the questions that a few of you have raised, and I apologize if I am going to get a little deep into some biochemistry and molecular biology, but I think it's useful to understand some of this ideas to answer your questions. So this is, of course, a pedigree that you might be acquainted with. This is a somewhat large one, not nearly as large as Dr. Sadoun's, but this is one of the pedigrees we saw in the late 70s. And, of course, each person that's filled in had sudden onset central scotoma, Leber's hereditary optic neuropathy. And all the affected people, of course, are related to the female lineage, but there's a predilection of males being affected over females. This particular family has the 11778 mutation. So as both of former speakers said, there are several things that are surprising about this kind of pedigree. The fact that everybody on the maternal lineage isn't affected, that males are preferentially affected, and something that they haven't touched too much on, and that is there are a number of different changes in the mitochondrial DNA that can predispose you to this problem, and that they themselves are somewhat different in their characteristics. So the questions then are, why are these different mutations giving the same clinical problem? So I would like to reiterate something that Dr. Sadoun said, but in a little bit more detail, and that is how does the mitochondria actually make energy? Because it's important to go a little deeper into this question if we really want to get some insight into the problem. And basically, this would be a mitochondrion within one of your cells. It has an outer membrane, an intermembrane space, and an inner membrane. And this is actually a symbiotic bacteria that got into the proto-eukaryotic cell. You're made of eukaryotic cells about 2 billion years ago. So inside each cell is a nucleus and a cytosol, and then thousands of these bacteria. And these bacteria generate the energy. So how does the bacteria generate the energy? Well, it takes it takes the energy in the food that you eat, and it converts it into energy forms that your body can use. So you might eat, say, a chocolate chip cookie or a muffin. That is actually a polymer of glucose. And your body then breaks those polymers into single units of glucose. Glucose then is processed by something called glycolysis. And then it generates pyruvate, which can then enter the mitochondria to, in fact, generate the energy. So the first thing that happens is the pyruvate enters the mitochondria, it goes through this cycle, and the purpose of the cycle is to take the hydrogens off of the hydrocarbons. So sugar is carbon, oxygen, and hydrogen. And the way that that hydrogen got onto that carbon was by plants. Plants split water using light energy, put the hydrogen on the carbon, and that's the plant sugar. So now your mitochondria is going to strip off those hydrogens and put that on a carrier called NAD. So now you have, instead of this oxidized form, you now have the reduced form with the 2 electrons and 2 protons. And now you're going to burn those hydrogens by the electron transport chain that Dr. Sadoun mentioned. So the hydrogens give up their electrons to complex 1, or NADH dehydrogenase. D is to remove, and hydrogen is removed. So it's an enzyme that removes the hydrogen. So the NADH dehydrogenase will then take 2 electrons, and it passes through a series of electron carriers in that enzyme, and then it hands it off to this lipid carrier coenzyme Q, which Dr. Sadoun mentioned can be used to treat mitochondrial disease, but is not very effective for problems of the brain because it's hard to get into the brain. But it carries the electrons along this lipid bilayer, which is the mitochondrial inner membrane, and hands them off to complex 3. And then complex 3 uses those electrons. It passes through a series of carriers, and then it hands it off to another carrier called cytochrome C. And cytochrome C moves it to the last enzyme, cytochrome oxidase, or complex 4. And that enzyme then takes the 2 electrons and adds them to an atom of oxygen and gives you a molecule of water. So basically, your mitochondria are just like a fire in the fireplace. They're burning hydrocarbons. They're giving off water and CO2. And so, in fact, it's a very simple, straightforward process that you use every day of your life. Now, that process of burning those electrons generates energy, but energy is very hard to store, and your body needs a lot of energy. In fact, no anatomical structure will move without energy. Newton said that mass won't move until energy acts on it. Well, you're the most animated thing in our known universe. Therefore, a lot of energy is flowing through your system to allow you to be active. And that is, in fact, the core of what we're talking about here. So how does that system then store the energy so that you can then do all the things that you want to do? Well, it does that by making a capacitor. So as the electrons flow through complex 1 and 3 and 4, the energy that's released is used to pump positive charges across the lipid bilayer, which is an insulator, into the intermembrane space. And that creates a capacitor that is acid on positive charge on the outside and alkaline and negative charge on the inside. So sitting in your chair right now are 100 trillion cells. You think of yourself as a single individual, but you're actually a colony of individuals. And within each of those individual cells is another colony of bacteria. And that bacteria, the colony, is the mitochondria. So sitting in your chair right now are 10 to the 17th bacteria, these mitochondria. And each of them is a charged capacitor, and that's 0.2 volts. So 10 to the 17th times 0.2 volts is the amount of energy of a lightning bolt. And that's the energy that drives everything that you do during your daily life. And the reason you eat breakfast, lunch, and dinner, and the reason you breathe is to maintain that capacitor. Okay? So the difference between being alive and dead has been known for thousands of years is when a person stops breathing. Well, why is that? Because once you no longer have the oxidizing agent to react with the hydrogen, the reducing agent, then you can't maintain the membrane potential. Once the membrane potential is gone, you have no potential energy to do anything, and your system stops, and that's what we call death. Okay? So, in fact, this is an essential process for the life process. Now, you might want to convert that potential energy into work. And the way that is done is to convert it into a chemical carrier called ATP, which Dr. Sadoun mentioned. And the way that's done is this membrane potential is used by this enzyme, the ATP synthase, and the protons flow back in across the membrane, and it causes this proton to spin like a wheel, and it binds then two substrates, ADP and phosphate, and drives the phosphate onto the ADP to give you ATP. The ATP then goes out to do work. And so then that phosphate can release that energy in running reactions, causing your muscles to move, or maintaining the membrane potential across the neurons, the axons that Dr. Sadoun mentioned. So basically then, your biology of energy is about oxidation and phosphorylation, and hence we call this oxidative phosphorylation. Now, there's a very important point to be made, and that is the efficiency by which calories in your food are converted into this capacitor, and the capacitor is used to make ATP, differs in probably everybody in this room. And we call that the coupling efficiency. You're coupling oxidation with phosphorylation. So some people are very efficient at burning calories and making ATP. We call them tightly coupled. So you'll get the maximum amount of ATP for the least number of calories. And those are the people that if they eat a chocolate cake, they'll gain weight. Whereas my wife is loosely coupled. That is, she needs to eat more calories for the same to generate the same membrane potential and to convert it into ATP. So she can eat lots of calories, and she wastes all that extra energy as heat. And so she annoys me by eating several pieces of chocolate cake and losing weight, right? So this is, in fact, exactly what's happening. Each of us is different metabolically. And those differences are critical to your predisposition to these various kinds of clinical problems, okay? So tightly coupled, loosely coupled is the ratio of calories, which is a unit of heat. A calorie is the amount of energy to raise 1 gram of water, 1 degree centigrade. So the number of calories you eat, that you burn, that's how much heat you put inside your body. So if you're very efficient, you need very few calories to do your work. If you're less efficient, you need more calories to do work. Simple. As Dr. Sadoun mentioned and Dr. Corelli mentioned, that if this system is inefficient, that is, if you inhibit the electron flows down this system, then the electrons build up in the system. They can't move through easily. And when they build up, they then sit there in the presence of oxygen. And that allows a situation to occur which one of those electrons can actually get misdirected from this electron transport chain and go directly to oxygen to give the first of the reactive oxygen species. That's called superoxide anion. But there are 3 of them: superoxide anion, hydrogen peroxide, and a hydroxyl radical. And all of these compounds want another electron, so they're oxidizing agents. And they damage the lipids in the membrane, the proteins that make the enzymes, and the DNA. So these reactive oxygen species are a byproduct of mitochondrial function. And if the mitochondria is inefficient or damaged, you'll get more of that byproduct. So the analogy is we're in Southern California. Analogies never work in Southern California. If you lived where I live in Pennsylvania, you would be worried in the winter about cold, and you would idolize a guy named Benjamin Franklin who invented the Franklin stove. Okay, so here we have my wife, and my wife is partially uncoupled. So she wants to eat a high-fat, high-carbohydrate diet, and if I eat what she eats, I gain weight like mad. Okay, so what's the difference? She's always hot and I'm always cold. So that's why in our bed we have 2 zones of our heating blanket, right? Never mind, that's going wrong. We don't need to go there. But anyway, the point is that here we are, we have our Franklin fireplace, and I'm always cold and she's always hot. So we have the Franklin fireplace, and I say, can we start the fireplace? She said, yes, don't put on too many wood. So I put on 1 log, and it's burning brightly. She goes out of the room, and I'm still called to put another log, right? She comes back, it seems a little warmer in here. She goes out and put another log. I keep doing this, right, because I'm still cold, and ultimately I put in too many logs. What happens? Suddenly the room fills up with smoke, and she knows I cheated, right? And that's because now there's too much reducing agent and not enough oxygen, and we get a lot of smoke. So that's exactly what we're concerned about here in this particular disease, okay? So this toxic smoke then will damage the mitochondria in the cells, and as you heard, it can trip this self-destruct switch called the permeability transition pore and result in the cell killing itself. Okay? So that's basically the idea. The mitochondria then makes most of the energy. It regulates something we call redox balance, which is very important for biochemistry. It makes these reactive oxygen species. It regulates calcium, which is important in your metabolism, and it regulates cell death. Okay. So what we have then is we have this small piece of DNA called the mitochondrial DNA, and it's inside the mitochondria. But to make a mitochondria takes thousands of proteins. So where are all the rest of the proteins? They're in the nucleus. So to make a mitochondria takes genes in the nucleus and takes genes in the mitochondrial DNA. So the mitochondrial genome are these thousands of nuclear genes plus thousands of copies of this mitochondrial DNA. So what does that look like? This would be your cell, this is your bacterial organism, and this is your nuclear cytosolic organism. Both of these have DNA. The DNA is replicated inside the cell, in the mitochondrial cell or in the nuclear cytosolic cell. Then the DNA is read into a working copy called messenger RNA, and the messenger RNA leaves the nucleus, or in this case, leaves the DNA from the mitochondria, and then it binds to these structures. They're called ribosomes. And ribosomes are little factories, and they convert the information in the DNA into proteins, and proteins give you structure and function. Okay? So this is really the magic of biology. How do you get a linear tape and make a three-dimensional organism? That's really something really special. So anyway, there's completely parallel systems between the nuclear cytosolic system and the mitochondrial system. But the mitochondrial system is different from the nuclear cytosolic system because it's still bacteria-like. So when it makes proteins, it puts a little tag at the beginning called N-formylmethionine, and that's the signature, I'm a bacterium. So, in fact, the mitochondrial DNA looks very bacterial-like, and the mitochondrial proteins are signed they're bacterial-like. And therefore, these products in the cell can be seen as if they're bacteria if they get out of the cell. Okay? So then what we have here is a system where if these escape into your bloodstream, you will mount an inflammatory response. So how do we avoid in a sick cell having these get out? We do that by having this apoptosis. What does the apoptosis do? It releases enzymes into the cell and it eats itself from inside out. And by doing that, it eats up all the bacteria. And so then their bacteria can't be released and can't create an inflammatory response. Okay. So this then is the mitochondrial DNA. It's 16,569 base pairs. It codes for these structural RNAs to make the ribosomes and these transfer RNAs that are involved in expression of these genes and 13 proteins. And they're called ND1, 2, 3, 4L, 4, 5, and 6. And those are for respiratory complex 1, cytochrome B for complex 3, CO1, 2, and 3 for complex 4, cytochrome oxidase, and ATPase 6 and 8 for complex 5. So what we have then is the mitochondrial DNA making 13 proteins, 7 of the 45 proteins of complex 1, 1 of the 11 proteins of complex 3, 3 of the 13 proteins of complex 4, and 2 of the 17 proteins of complex 5. Now here's a very weird thing. If in fact the original bacterium transferred 1,000 to 2,000 genes to the nucleus, why the heck did it keep 13? Okay, so the answer to that I think is very clear. Those 13 proteins are the electron and the proton wires for complex 1, 3, 4, and 5. And what do they share? The capacitor. So it's just like an integrated circuit. Every component of the integrated circuit has to be balanced with every other one. If one of those enzymes became leaky for protons, it would short the capacitor and you would be dead. Okay? So how did Mother Nature make that happen? Mother Nature put all of the electrical circuitry on a single piece of DNA. And then she made that piece of DNA inherited from the mother. After all, she was Mother Nature. So anyway, the mitochondrial DNA is transmitted from mother to all of her children and from her daughters to their children. But when the male mitochondrial DNA enters the oocyte, it is seen as foreign and is selectively destroyed. So women have thrown men out for 2 billion years. It's really appalling. Okay. So anyway, nothing has changed. Now, inside each cell, there are thousands of these mitochondrial DNAs. And they are constantly growing inside your cells all the time. They're replicating, and at the same time, your cell doesn't want too many of them, so it has a system to eat them up. So they're in a steady state. And you make more mitochondria, and they're eaten up at the same rate. But that means that as the mitochondria replicate their DNA, they can make mistakes. Those are called mutations. So inside your cells, this replication process can accumulate mutations, either in the female germline to give new mutations to give labors, or even into all your cells right now. And so someone as old as me, I have lots of mutations in my mitochondrial DNA. That's why I look so bad. Okay, so anyway, we call this situation heteroplasmy. That is, you have a mixture of normal and mutant. So the mutant will just show as red, and the normal as blue. Now, this is interesting because in this heteroplasmic oocyte that just got fertilized, male mitochondria was thrown out. If it divided down this middle, then both cells would have some mutant and some normal. But if it divided this way, then this cell would have only normal mitochondrial DNA, and this would have twice as many mutant. So what's unique about the mitochondrial DNA is that a heteroplasmic cell can segregate during the development of an individual. So that you could start out with, say, 50-50 mutant and normal mitochondrial DNAs, but by the time you became an adult, you could have some tissues that have predominantly good mitochondrial DNAs and others that had predominantly mutant. So now this gives you a very complicated situation where everybody, by chance, can have a different distribution of mutant and normal in heteroplasmy. And that's one of the reasons why we get a lot of variability in mitochondrial disease. Okay. And as the amount of mutant mitochondrial DNAs increases, it passes below the minimum energy for that particular tissue. And once that happens, then the tissue begins to malfunction, and that gives you a phenotype. And Dr. Sadoun mentioned why the optic nerve might be particularly sensitive to this, because it can't have any myelin, and therefore it needs a lot of mitochondria to maintain those membrane potentials. So that's a high energy demand tissue. Okay? All right. And the mitochondria is bathed in oxygen radicals, and that's a potent mutagen. So you're getting a lot of mitochondrial DNA mutations. So there are three classes of mitochondrial DNA mutations that are germane to mitochondrial disease and possibly labors. The first class, of course, is what you've all heard about. That is a mutation that occurred in the female germline and was transmitted to the children. And that mutation, let's say the 11778 or the 3460, which is up here, or the 14484 mutation, all complex 1 gene mutations. Once you have a pure mitochondrial lineage, then everybody in the female lineage will get that same mitochondrial DNA. On the other hand, if perchance this was a new mutation, N11778, and these actually, there are quite a few people that have this, then that family might be heteroplasmic. And now you might have some children that get almost all mutant mitochondrial DNA, and by chance others that would get less. And you'll see a pedigree like that in a minute. Okay, so we then can have these maternally inherited mutations, which then predispose you to these diseases. But there's another class. Yes, ma'am. No, that's perfectly fine. No, not at all. It's not dumb at all.
In the remedial group.
Yeah. Well, if the mutation is in your family and it's all the cells mitochondrial DNA are mutant, we call that homoplasmic, right? All the mitochondrial DNA are mutant. Now, that female line will always have the mutant because the only thing you can inherit is mutant mitochondrial DNA. But that mutation arose sometime in the past. Not this mutation that an individual has wasn't in your maternal lineage all the way back to Eve. It arose at some time. And at that time, there were a few mutant mitochondrial DNAs and all these thousands of normal ones, right? There was a time when there was just a few mutant and thousands of normal. And then, in certain cases, those mutant mitochondrial DNAs become enriched to give you a maternal lineage with that mutant mitochondrial DNA. Okay? Okay? No, that's great. Please do ask questions, because I understand this is tedious, but one of the questions that people ask is, what is the background variation and what does that have to do with the disease? And I'm going to try and explain that to you. Okay. So, mutations are arising all the time. We saw in our clinic, we don't see very many Leber's patients now, but we used to see quite a few, and a certain percentage, I think about 20% of the families we see have a heteroplasmic mutation rather than a homoplasmic mutation. And that, if your family has that, then there is a new dimension to the expression of the disease. One of the families we're working on right now is pure for the 11778 mutation and heteroplasmic for the 14484. So they have a predisposition of males because of the 11778, but when the 14484 gets high, then the women get affected. Okay? So this is just an example of how you could have this interaction. Okay? Okay, so in addition to these maternally inherited maternal lineage mutations, you also have ancient polymorphisms. And everybody in this room has a slightly different mitochondrial DNA sequence. And these mitochondrial sequences go back tens, hundreds of thousands of years. And they correlate with the geographic origin of your ancestors. So in fact, if you were an African American, for instance, then 3/4 of all African Americans have a polymorphism in this ND1 gene, and that we call macro group L. We just call it letter number L, that variant for sub-Saharan Africans. This variant we call H, that's found in half of the people in this room, half of the Europeans. A variant A, B, and C, and D, those arose in Asia, and they allowed women to cross the Bering Land Bridge and colonize the Americas. So these polymorphisms changed people's metabolism to allow them to live in different environments. Okay? And then the last point is you accumulate these somatic mutations because your mitochondrial DNAs are replicating all the time inside your cell. Okay, 3 classes. All right. So then the next thing you need to understand is what Dr. Sadoun said, and that is different organs rely on mitochondrial energy for different extents. So the optic nerve, just at the point where it leaves the retina, that's highly energy intensive. In general, the brain is the highest energy demand, 2% of the body weight, 20% of the oxygen. So the energy utilizing tissue, the brain is at the top. But heart, muscle, renal, and endocrine systems are also very high energy demand tissues. So in fact, people with more severe energy defects than the classic labors can also have additional symptoms. So one of the things that Dr. Nikoskalainen said, which she was mentioned earlier in her earlier work, is that some labors patients also have cardiac conduction defects, slight changes in heart rhythm. Now, some people would disagree with that, some people agree with that, but that is possible. Okay.
For example, AFib?
Yeah, so you might get cardiac dysrhythmias, or you could, well, I'm going to have to defer to whether you could ever go to the extreme besides cardiac conduction defects, whether it would become serious enough to get to that level. But you can sometimes, on some labors patients, see slight changes in heart rate, heart beat. You're going to, if you want to clarify that, please say.
Well, yeah, it's exactly as you say. You may have atrioventricular partial block.
Right.
There are conduction usually.
Yeah, they are not generally life-threatening, but if you look carefully enough, you can see other examples. Or Dr. Corelli mentioned that if you take a labors patient and ask her to exercise, then you'll see slight differences in her muscle energy output from, say, another person. That doesn't mean that that has affected his or her quality of life, but it is a measurable change. Okay? So what we have is a systemic energy defect. Just a moment. But the organs with the highest energy demand are going to be the ones that are affected. Yes, ma'am? Incomplete bundle block. Yeah, again, these are the same. So the heart has an electrical pacemaker, and that pacemaker is using energy from the mitochondria to fire the membrane potentials around these cells. And these cells then, when they depolarize, they then send a spreading signal into the heart to cause the heart to contract. So the pacemaker is very sensitive to energy defects. And so more severe mitochondrial disease will have, in fact, even more severe cardiac dysrhythmias, as we call them. Okay? But this is not usually a life-threatening issue in classic labors, although some individuals in this room have more than classic labors, and they might have then some of these additional symptoms. Okay. Well, there are also tissues to store energy, to make ATP. There's tissues to sense, maintain your blood energy level. The liver maintains your blood glucose level because that's the source of your calories. And the pancreas senses levels of calories and adjust your metabolism depending on the availability of your food sources. So our center is very interested in the possibility that a lot of different kinds of clinical problems might in fact have an energetic defect. And so here we have the idea that energy is central to everything that's important to being alive, and you could change energy output by having alterations in nuclear genes, as was mentioned, by changes in the mitochondrial DNA, either deleterious mutations or these ancient polymorphisms. But also the mitochondria is where you burn all your calories. So depending on what you eat and the demand of those of your energetic needs in the day, those could also affect mitochondria. And the mitochondria is extremely sensitive to lots of different toxins. It's your Achilles heel. So in fact, many industrial chemicals are toxic, as you heard from Dr. Sadoun. Many agricultural chemicals are very toxic to the mitochondria. Rotenone is a common rat poison. Paraquat is used as to kill off plants. All of these are potent mitochondrial inhibitors. So one of the things that some of us are concerned about is that we add more and more of these things to our environment. None of them are so toxic as to kill you, but they're lowering your energetic threshold and making you more prone to these clinical problems. And that's what Dr. Sadoun mentioned with the smoke. Okay. So what would be, if you had this energy deficiency, what would happen? Well, depending on the amount of the energy deficiency, you would first get very subtle brain effects, optic atrophy. But if it's more severe, you might get a heart defect or a renal defect or a muscle defect. So the more severe the defect, the more tissues are affected. And then, in fact, of course, since this is how the calories flow through your body, it's going to affect things like diabetes and obesity. Like my wife will never get fat, and I will always be fat. And that's just really annoying. Okay, so then you have these thousands of bacteria inside your cell. And if the cell breaks open, those bacteria are released. Well, you'll get an inflammatory response. So there's often inflammation associated with severe mitochondrial dysfunction. Okay? So basically then, because the somatic mutations accumulate with age, that gives you the aging clock. And the older you get, the less your energy output is, and you then have more and more symptoms in all these energy-sensitive organs. Okay, so that's the basic idea. Okay, so this is a family that is a case of a heteroplasmic mitochondrial DNA mutation that gives labors. And this mutation is at the nucleotide position 14459. It is in the ND6 gene, and it just changes an amino acid from alanine to valine. Well, this pedigree then is fairly large. It's actually the first maternal pedigree we studied after we found a maternal inheritance of the mitochondrial DNA. And what you can see is all the colored symbols are ones that had a clinical problem. And the beauty of writing a pedigree for a mitochondrial DNA mutation is you don't have to talk to any men. And if you're a man, that's very nice, because you only talk to women. It's very nice. So here you can see that these people are related through the female lineage, through this woman, to all of these people. Okay? And this woman had this daughter who had 3 husbands, and there are some affected individuals in red. This woman had 2 husbands, and there are some red affected and blue affected individuals. This one has both. Here's a pair of twins, one that's red, one that's blue. They both had 2 husbands, and they have affected children. So when I first found this pedigree, I was very excited because I thought this is the first case of a mitochondrial DNA disease. And what do these people have? These people in blue have classic Leber's hereditary optic neuropathy. They were fine, sudden central vision loss, and then blind. But what was astonishing is that these people had what's called generalized dystonia. And what is that? This is a lady trying to get out of her wheelchair, and she's rigid. So it's like a early onset Parkinson's disease. And that's associated with this region of the brain, the cells dying out. And that's a portion of the brain that coordinates motion, called the basal ganglia. Okay? So what we have here in this pedigree is some people with Leber's and some people with this very severe neurological disease. So I knew about heteroplasmy, and I wasn't bothered by this. So I ran to my neurology friends, and I said, neurology friends, said, Eureka, we have a maternally inherited neurological disease, therefore mitochondrial disease exists, because at the time nobody believed that is the case. And they looked at the pedigree and they said, well, that's all well and good. These people have dystonia, but these people went blind. And we never go to parties with people that see people that go to blind, so it can't be the same disease, and it's not maternally inherited. I thought, gee, that's a real problem. So I went to see an ophthalmologist, because all my ophthalmologist friends are much more liberal. And I said, Eureka, we have a maternally inherited ophthalmological disease. And they said, well, just a minute now. These people are seeing the neurologists, and we never talk to them. It can't be an ophthalmological disease, and it's not maternally inherited. So I was so frustrated. How could I tell people that maternal inheritance was important? Then I realized, looking at this pedigree, see this woman with multiple husbands, this woman with multiple husbands, these women with multiple husbands. Eureka, a maternally inherited disease is having multiple husbands. Okay, well, anyway, that did not help me in getting anything published, but it made me feel a lot better. What in fact is happening here is that this is a new heteroplasmic mutation. And these individuals have some normal and some mutant mitochondrial DNA. So that's a milder defect. But as time goes on, the normal mitochondrial DNA got lost, and then these people are pure mutant mitochondrial DNA, and then that was a more severe biochemical defect, and then they got a worse disease. Okay? So this is just an example of how a new mutation can give you variable expressivity. Now, this particular change changes this amino acid that is conserved in human, cow, whale, mouse, rat. This is a kind of frog, sea urchin. And so this is a pretty severe biochemical defect, whereas the 14484, the mild reversible one, that is very mild. It only affects changes in amino acid found in human and cow. So you can see now we have a differential. The severe mutation only gives you labors when it's heteroplasmic. It gives you a different disease when it's pure mutant, because it has a more severe biochemical defect. Whereas the milder mutation only gives you the blindness when it's pure mutant, because it's much milder. So you need more copies that are mutant. Is that clear? Okay. Yes? No? I'm going to have a Coke. So, yes, ma'am. Where's a male-to-male transition? This woman had this husband, and then each of them, each of those marriages had multiple children. To the left of her. Here? Here? Well, this male was affected, but none of these people were diseased. Only the people that are either red or blue had a disease. Okay. I filled this in so you could see it from the back. But so, unlike a classic one where clear would be unaffected and colored would be affected, here I had two different phenotypes. So I made one blue, one red, and then everything else I just filled in so you could see it. Okay. So only men and women on the maternal lineage were at risk for the problem. Okay? And so depending on their percentage of mutant and normal mitochondrial DNA, they either got a milder disease, that is optic atrophy, or a more severe disease, basically a very severe, ultimately lethal neurological disease. Okay? All right. So again, then the severity of the mutation, the very severe mutations, will give you the same enzyme defect if half of the mitochondrial DNAs are still normal, because that's accommodated the severe mutation. Whereas the very mild mutation gives you that biochemical defect only when all the mitochondrial DNAs are mutant. Okay? Yes, ma'am. Yeah. So if we want to know how important an amino acid is so let's take the ND6 protein, and it is encoded by DNA, and each 3 nucleotides of the DNA makes an amino acid. Okay? So a protein is made of a string of amino acids, and there are 20 amino acids. So what we have here is an amino acid called alanine, and that alanine in this portion of the protein for ND6, it got mutated, that DNA got mutated to give you a new amino acid valine. And that altered amino acid from alanine to valine, that's what caused the biochemical defect that gave the disease. Okay? So how can we know how important that alanine is? If the alanine were random inside the protein, that it could be any amino acid, then changing it wouldn't have made a difference. But if that amino acid is found in all animal species exactly at that place, then it must be important because all the animals had to have it that way. So what we've done is aligned all of the amino acids according to animals from human all the way down to worms. And we've asked, is that amino acid always found in that position in lots of different organisms? And the answer is, it is found in that position all the way down to sea urchins, which is a long time ago, about 500 million years. Therefore, that amino acid in that position is important, because if it weren't important, it would be random. Okay? On the other hand, look at this one. This is the 14484 mutation. Well, it's methionine, that's an amino acid, only in mammals. Even in some mammals. In the whale and the rat and the mouse, it's leucine. So this amino acid is less constrained than that one. So that mutation will have a less biochemical defect. Okay? Oops. Okay, moving right along. Anyway, what we see is that homoplasmic people are sicker than heteroplasmic people for this severe mutation. That's the important point. So back to this case. Now here we have a mutation that is homoplasmic, pure. So everybody in the maternal lineage will have that mutation, right? The question is, why is there variability in the phenotype, and why do different mitochondrial DNA mutations give the same phenotype? These are two questions. Okay. So this is just a list of some of the mutations that have been found in the mitochondrial DNA that cause this kind of blindness, Leber's hereditary optic neuropathy. This one you've heard about, 3460, 11778, 14484. They're the common ones in Europe. But this one, 14459, is the one we just talked about. That's the heteroplasmic one that gave you the different phenotypes. And there are these milder ones. This is 10663. So what's remarkable is if you now look at the enzyme, complex 1, that all of these genes are involved in, the severity of the enzyme defect is different with the different mutations. So that's kind of weird, because you would think that to get this blindness, you would all need the same biochemical enzyme defect. But here we have different mutations, different enzyme defects, all giving the same problem, blindness. So how could that be? The answer is that there's another factor. Now, Dr. Sadoun and Dr. Corelli said, well, maybe there could be environmental factors, like toxins, that would make this more penetrant. That's fine. But there could be genetic factors that also make it more penetrant, and that turns out to be very important. So, there is something we call haplogroup J, and it was on Dr. Sadoun's pedigree in Brazil. And haplogroup J, when it's matched with one of these somewhat milder mutations, it increases the probability of your going blind. Okay? So the most severe mutation, it doesn't matter what background you have. It's the biochemical defect is so severe, you'll always go blind. 3460 is milder. It's found in about 10% of all cases have this J. Well, 10% of this population is J. Okay? This mutation, which is milder, a third of everybody is J. This mutation is even milder, 3/4 are J. And this very mild mutation, everybody's J. So therefore, this J background is somehow modulating the sensitivity of the mutation. Does that make sense? I'm going to tell you all about J in just the next slide. This is another variant, 3394C in ND1. It's very rare, causes a mild effect, but it also modulates some of these milder mutations. Okay. So what are these lineages? So the mitochondrial DNA has been inherited from mother to daughter, all the way back to mitochondrial Eve, which is about 200,000 years ago. The only way it can change is by sequential mutations. So every time a new mutation occurs, there's a branch in the tree, right? Because there's with the mutation, without the mutation. So if I sequence the mitochondrial DNA of everybody in this room, and I compare the number of nucleotide differences between your two mitochondrial DNAs, the number of differences would be proportional to the time you shared a common mother, right? Because those are accumulating along the lineage. So what I can do, or what we have done, is sequence the mitochondrial DNA from indigenous people from all over the world, and then simply compare their mitochondrial DNAs. And then knowing where those people lived and knowing their mitochondrial DNA relation to all other mitochondrial DNAs, you can reconstruct how all those people got there. You can actually reconstruct the ancient migration of women. Okay? So what we've been able to do is reconstruct the history of women. So this then allows you to realize that mitochondrial DNA started in Africa about 150 to 200,000 years ago with what we call L0, because all the African lineages we call L. Okay? So L0 is a group of mitochondrial DNAs that are at the very foot, the base of the tree, and they're found in the Khoisan people of the Kalahari, the African bush women and men. Okay? So classic ancient population. Then that lineage gave rise to two lineages, L1 and L2. Those are different pygmy populations in the Congo. Then that gave rise to lineage L3, which is all the Bantu populations. Okay? Now, L3 gave rise in Ethiopia to two mitochondrial DNAs, M and N. These were the only mitochondrial DNAs that left Africa and colonized all the rest of the world. All these other mitochondrial DNAs stayed put, but these left and colonized the world. So M stayed in the tropics and moved all the way down to Australia. Ultimately, later, it moved up into the temperate zone. But N did something quite remarkable. It went right north. And first, it gave rise to European lineages we call IWX, and it gave rise to an intermediate R, and then European lineages H, J, T, U. These are just lineages we discovered over the years. Okay? So the important point is that M stayed in the tropics, and M went into the cold. Okay? So why would this be relevant? Because here in the tropics, if you want to be a woman and reproduce and survive, then you need to be able to run away from lions. Well, how do you run away from lions? You need to use all your calories to make ATP. You don't want to waste it to make heat. So you're going to burn those calories, you're going to make that amount of heat, but all of that was also used to do work. So you're very good at running, and you're tightly coupled. You're most efficient. But up here in the Arctic, the lions froze to death. That wasn't the problem. Your problem is you're going to freeze to death. So how do you avoid freezing to death? You acquire mutations in your mitochondrial DNA that decrease the efficiency. Now what happens? You have to eat more calories for the same amount of ATP. But calorie is a unit of heat. So the more calories you burn, the more internal heat. So those women are more resistant to cold stress because they're generating more internal heat. Okay? So the mitochondria has changed by latitude, allowing people to live in different climatic zones. And it's done that by changing the mitochondrial efficiency. Why is that so important? Because if you add a mitochondrial DNA mutation on an inefficient system, you're going to have a bigger biochemical effect. Whereas if you add the mutation on a more efficient system, it's going to ameliorate the effect. Therefore, lineage J has a greater effect on the labor's mutations than lineage H. Okay? So when you say, someone says you have a 11778 mutation on haplogroup J, they're giving you critical information about how that background is modulating that effect. Okay? All right. All right. So what's the evidence for that? Well, here is out of Africa. This is the M lineage that stayed in the tropics, and it doesn't have any changes that affect function. But this is the N lineage that went into the temperate zone where it's cold, and it acquired 2 amino acid changes in mitochondrial DNA-encoded proteins. And what did they do? They decreased the membrane potential. That is, they increased the heat production. Okay? So this was the adaptive mutation that allowed people to leave Africa. So why didn't all those L mitochondrial DNAs leave Africa? Because they're not adapted to the cold. They couldn't go there. But this N could. Okay? So what's the biochemical effect? Complex 1 is what's defective in labors. So if we now look at N and M, and we're going to look at the enzyme activity of complex 1 with this very sensitive assay. Here are the N lineages, different ones. This is F1 and B. They're just given names. And you can see with this particular 3394T variant, the T variant has more complex 1 activity than a C variant on the exact same background. Or here, B4, here's the T variant, here's the C variant. Well, the C variant increases your penetrance for labors. So if you've got that plus the mutation, then you have a bigger effect. Okay? So these kinds of lineages have been now correlated with a whole range of neurological diseases, metabolic problems, and inflammatory diseases, aging, cancer, even athletic performance. So energy is important, and the mitochondrial DNA defines the energy. Okay, so the last point I want to make then is how can I prove to you that this is really important? And the way we did that is a group of us, including Alfredo and Valerio and their colleagues, we made a mouse. Okay? We made a mouse with a mutation. Now, what mutation would we like to put in? We'd like to put in a mutation that gives optic atrophy because we want a Leber's mouse, right? However, you live, hopefully, 100 years. The mouse lives only 2 years. Therefore, since it takes 25 years for you to go blind, if you're going blind, then the mutation in the mouse would have to be more severe to go blind in 1/10 the time, right? So our goal was to find a Leber's mutation that was more severe than the common 11778-14484. Okay? So this family was found by Massimo Zeviani, which, of course, according to Valerio, if you live in Italy, you have to collaborate with. I did not collaborate on this particular pedigree, but this is very interesting because this woman was heteroplasmic. She had half of this mutation and half not of this mutation. And this mutation is in the ND6 gene, and it changes this proline, which is highly conserved over all these animals, to a leucine, codon 25. This woman then had the optic atrophy, but through her sister, who had 3 different husbands, she had these 2 children from this husband, this child from this husband, and this child from this husband. All of these children died of a very severe childhood disease called Lee syndrome. It kills them in their first 5 to 10 years of life. And this child had less severe disease, but still much more severe than this lady. Well, what's the difference? These people were 50% heteroplasmic. These were all pure mutant, and this had some wild type. So now we're seeing the phenotype graded by the mutation. So if we could make a mouse with this mutation and it was pure mutant, then we might get the blindness in 2 years rather than you're having to wait, 25 years. Okay, so that's what we did. So we found we isolated a mouse cell line that had this exact mutation. So how are we going to put those mutant mitochondria into a mouse? They're already in a cultured cell, growing in the cultured cell. So what we did is we actually physically removed the nucleus. So now we have the cytoplasmic fragment with its mutant mitochondrial DNAs. Then we took a mouse and we took its early embryos, which have cells that can make all the cells in the body. They're called stem cells. We took the stem cells, female stem cells, and we took the female stem cells, removed their mitochondria by a drug, and put in the mutant mitochondria. So now we have the stem cell nucleus with the mutant mitochondria. We then put it into a foster mother, and we get an animal made from these stem cells. And it's picked up the mutant mitochondrial DNA. Okay? So now what we've done is put the same mutation that's found in a human that gives optic atrophy into the mouse. Okay? So then what happens to the mouse? Well, these mice get a slight reduction in their electroretinogram result. That is the way that the eye responds to light. And according to Dr. Sadoun's excellent reviews, if you look at some Leber's patients, you will see this small diminution in electroretinogram response. So that's not inconsistent. But this is the beautiful data that came out of Dr. Sadoun and Valerio's laboratory, where they actually now looked at the optic nerves of these mice. So on the left is a normal optic nerve, a normal mouse, and this is the mutant mouse. These two have exactly the same nucleus. They only differ by a single nucleotide change in the mitochondrial DNA, in the ND6 gene. And you can see, just as Dr. Sadoun mentioned, the axons have become swollen. Okay? And in fact, it's the small caliber axons that selectively are lost in these mice. So here in red are the axon sizes of the control, and here are the axon sizes of the mutant. And you can see that we're losing the small fibers and retaining the large fibers. And that gets worse as the animal ages. Okay? So that's just like Leber's. Okay. if we now look at the optic nerve, and this is a very beautiful picture that they sent me, just as Dr. Corelli showed you, you see in some of the axons the thinning of the myelin sheet. And you see the accumulation of these damaged mitochondria. So there are more damaged mitochondria, and they're associated with optic atrophy. So why would having a mouse be important? Because now we can ask, what's the biochemical defect that causes labors? Remember, the question was it energy? Was it reactive oxygen species? Was it apoptosis? That was what was posed. So now we can actually go to the brain of an animal with a Leber's mutation, and we can say inside the brain, what's the biochemistry? Okay? Now, if you just isolate the mitochondria and then study them, they're out of context. That isn't what's happening in your brain. So what we did is we took the mouse, we homogenized the brain, and we isolated what's called synaptosomes. So here's the axon, and this is the little end that's going to communicate with the other neuron. So by homogenizing the brain, this pinches off, and now you're giving this little bit of a cell with just its mitochondria. So now we can really study the mitochondria inside a brain cell. Okay? That's really important. So the first thing we did is ask, well, did this mutation cause a change in the ATP level? And the answer is no. There was no decrease in ATP. That was a big surprise to me. But when we asked the brain cell to now generate more energy, what we found is as more energy demand was made on the cell, the oxygen radical production went way up. So what does this say? This says that it's the oxygen radicals that are killing off the neurons, okay? Not the ATP. Well, that's really important because phytoestrogens are going to increase the antioxidant defenses, and that's probably why they're working so well. Okay? So this then indicates where we need to go for therapy. So all of this discussion then is to indicate that with this mouse model now, our collaborative team of Dr. Valerio Sadoun and ourselves, we now can take this mouse, treat it with different environmental toxins, find out which ones cause the phenotype to get worse. But moreover, we can treat it with phytoestrogens, we can treat it with gene therapy, and we can tell you what is really going to work before we try it on you. So this mouse took 20 years to prepare, but now we're in a position that we can really begin to understand therapeutics, and that was the whole point. So, of course, I want to thank a lot of people. There was a whole box here that said Alfredo Sadoun and Valerio. I don't know. Sorry, guys. But anyway, we had a lot of great collaborators on this work. So thank you very much. Yes.
Mutation stable generation to generation?
Yes. Because it's homoplasmic, which, again, in the humans, it was heteroplasmic. And because they had 20 years for the cumulative effect of what we think was oxidative damage, they only got optic atrophy in 20 years when they had a partial enzyme defect. But for us to get the defect in 2 years, well, it's actually 1 year, then we needed a pure mutant, so we had a higher oxidative load, so we could get the same effect on the cells in a shorter period of time. So this is homoplasmic, and the beauty of it is it absolutely breeds true. So now we can make hundreds or thousands of mice and compare identical animals with different environmental challenges. Yes, sir.
Suppose some human carriers have consumed more antioxidants or environmental issues like that, as you mentioned. Would that be determinative on, or the correlation on whether that person gets or converts?
Yes, so that's a really, really good question. I would hypothesize that the kind of diet and the kinds of, we'll call them drugs or chemicals that you could take, could modulate this oxidative stress situation. For instance, the phytoestrogens would increase the antioxidant defenses, and that would then decrease your risk. Now, there's a problem here in the general literature, And that is, people call things antioxidants as if all antioxidants are the same. Okay? So one of the common antioxidants is vitamin C, vitamin E, beta-carotene. So if you read the health literature, and I try to read it pretty carefully, you'll often see, well, we did a epidemiological study, and we looked at people that had and took these antioxidants and those that didn't, and generally they found there was no difference. Well, why isn't that working? Because these particular compounds are only briefly antioxidants. That is, their job is to take an electron, pick it up, and then hand it off. So they are in balance. They're more like buffers. So if you have a chronic oxidative stress and you take those antioxidants, as soon as they get into your cell will drive them toward the oxidative side. And so they do not change your metabolic status. So what we've been developing is drugs that go into the mitochondria, and themselves are catalysts so that they eat up all the oxygen radicals. Now, they are literally changing the oxidative stress, and those have remarkable potential therapeutic effects. So don't be confused when someone says, well, we did a survey of all people with 11778 and whether they took vitamin E and vitamin C, and we saw no difference. Yeah, but that wasn't the right antioxidant to use in the first place. Okay? So I really do believe that there will be antioxidant therapies such as idibidone and EPI 743, which will be beneficial because they are acting much more directly and in an assertive way on this oxidative stress.
Two different classes. So the class of green tea, C, E, and whatever, those are different classes.
Yeah, although I'm actually a big fan of blueberries and green tea, so I'm not going to argue that some of these very, very good nutrient supplements could be helpful. We've just never had a model to test it. But now, hypothetically, we could take these mice and put them on a green tea or a blueberry diet and ask if you had a very high load of those kinds of phytoantioxidants, which, by the way, are very potent, they might, in fact, be beneficial. We did a study a number of years ago on ginkgo biloba, which is a very classic herbal medicine in the East. And we fractionated it into all its high molecular compounds, and several of those high molecular compounds were extraordinarily potent mitochondrial antioxidants. So, in fact, Mother Nature cares about this, and plants have found ways to help with this antioxidant, hence the phytoestrogens. But we just have to be careful not to put them all into the same bailiwick and then throw out the baby with the bathwater. So just because it's a bad experiment didn't give the result doesn't mean a good experiment won't be helpful. But you have to give us time. It took 20 years to make the maps. Now we have to do the studies.
Thank you.
Thank you all. Oh, yes, one lady here.
When you're tested for the LHON mutation, you find out at that time whether you're homoplastic or...
Yes, ma'am. When they do the test, they should say it is a homoplasmic mutation or it is a heteroplasmic mutation. And
Even 10 years ago?
well, if you were homoplasmic 10 years ago, years...
These people, they go through cycles. The pheno labs for a couple of years
I'm
to
sorry.
give you the results.
Right.
And then you have to write to them and say, but what is it? Oh, well, you really want to hear it is. So they
Well,
know
yeah.
it, but they don't always report it.
Yeah, I mean, if you have a good clinical report, which has done the experiment in the right way, you should get factual information that will tell you whether you're heteroplasmic or homoplasmic. But understand that this disease mutation, the first one, was only found 25 years ago. There's the whole methodology of determining these things is very new. And so there's actually not very many labs, besides, say, Valerios, that really can do this in the right way, or Alfredo's collaborators. Yes,
Do
ma'am.
I understand right that if you are homoplastic,
That's right.
then that increases, like, say, if that's what my testing at, does that say increases my children's risk of actually, or my risk of becoming blind?
If you're homoplasmic, you are genetically at the maximum risk for that particular mutation. But let's say the mutation were the 14484.
which is the less biochemically severe mutation, your then risk of you and your maternal relatives would be lower than if you had the 14459 mutation, okay, which is a very severe mutation. So there's effects of genotype, there's effects of heteroplasmy, effects of environment, and also effects of nuclear genes. So this was the variability that Dr. Sedin and Dr. Valerio were saying, Corelli, that we can't yet give you a personalized medicine prediction of a individual's risk because there are enough additional factors right now that we don't know that we can't make that prognosis. We would like to be able to do that. That's the ultimate goal. And as he said, seeing in the retina telangiectasia. This is my third lecture today, so